So as we have participants from 58 countries, let me wish you a very good morning, afternoon, or evening. It's really a pleasure to welcome you to our 8th session of our ILSI annual symposium. And today's session addresses COVID-19, nutritional vulnerabilities, and food supply challenges. So we have a great program today. We have a session for 2.5 hours. And before we start by introducing our co-chairs, I want to take a minute to recognize the work that all the members of the planning committee have put for almost a year. We had several meetings to ensure we are looking at topics that would be interesting to the audience. I think we have covered many topics, whether it's nutrition, the food system, sustainability, or food safety. Again, as you can see on the list of on the slide, we have diversity from different parts of the world. So I think we were able to put up a very, very nice program with 33 speakers. All showed up, which I think also is quite a success. We're quite happy, and the 12 co-chairs. So before we kick off, I just want to say that the views expressed in the following for presentation are the views of the speakers and do not necessarily represent the views of the International Life Sciences Institutes. And I want also to mention that each speaker had the total freedom to prepare his presentation. So let's start by introducing our first co-chair, Geoffry Smith. Geoffry is the chairman of the Essential Micronutrient Foundation, a non-profit organization which addresses micronutrient deficiencies globally as a public health issue. He is also the director of Nutrition Strategies International, which deals with food and nutritional issues in developing countries. Last and not least, Geoffry Smith is the president of ILSI Southeast Asia region, based in Singapore. So, Jeff, the session is yours.
Okay, thank you, Stefan. It's a great pleasure. Welcome everyone from around the world. Just let me give some very brief contextual remarks on, I think, this important topic and the excellent speakers that we have. So just to give a sense of this this topic, it's very easy to overlook in the past decades the dramatic decreases in infectious disease mortality over the past century. So just to give an example, from 1900 to 1980 in the US, deaths from infectious diseases dropped by 95%. More recently, developing countries saw a similar trend. Infectious diseases in Thailand, for example, fell by 75% from 1958 to 2009. Even more recently, global deaths from measles fell almost 90% from 1990 to 2019. Nevertheless, infectious diseases remain a serious problem, as we've been dramatically reminded over the past year. Estimates prior to COVID-19 were about 15 million deaths annually from infectious diseases globally, with respiratory infections accounting for about a quarter of those. COVID-19 so far has caused an estimated 3 million deaths, and of course has increased concerns about future pandemics. Today we're going to look in part at the potential impacts of nutrition and ability to deal with infections, especially viral infections. So there's a lot of agreement, a lot of good science showing that a wide range of nutrients, including not only vitamins and minerals, but also essential amino acids and fatty acids, are essential to support the immune system to operate optimally. To put it another way, the immune system is adversely affected by malnutrition. We've identified specific functions of specific nutrients and immune response, but there are still fundamental issues that are not resolved. These include appropriate levels of specific foods or nutrient intakes to prevent specific individuals or populations from specific diseases, the exact role or response of the microbiome to food and nutrient intakes in signaling immune response, and the effect of aging and other conditions such as obesity or diabetes to affect immune response and how those are influenced by nutrition, and the general impact of nutritional status on immune response and even response to drugs and vaccines. It's perhaps worthwhile to note that the term immunometabolism, the study of these metabolism links and interactions between nutritional status and immune response, was only coined in the past decade. And to add to these nutritional issues, it's clear the current pandemic impacts have included disruption of the food supply in many countries. Today, we have an excellent group of speakers to address these questions. We'll have 4 presentations, followed by a panel discussion, an opportunity for the audience to pose their question to these experts. So I encourage you to enter your questions into the question box that you have on GoToWebinar, and we will be able to see those. Our intention is, unless there's a burning question, we will go through all 4 presentations and then deal with the questions in the panel discussion. If there's something very urgent for a particular presentation, we can take that up. So let me introduce quickly the first speaker, my co-chair, Dr. Fülöp Calder, professional of nutritional immunology at the University of Southampton in UK, where he's also department head of human development and health. His work includes more than 700 papers, many on nutritional modulation of immunity, inflammation, and cardiometabolic disease risk, with an emphasis on fatty acids, especially omega-3 fatty acids. His awards are too numerous to mention, but he's a fellow of the Royal Society for Biology and the Association for Nutrition. He's also a leader in nutrition as a past president of the Nutrition Society of the UK, past president of ILSI Europe, and currently president of the Federation of European Nutrition Societies, FENS. He was editor-in-chief of the British Journal of Nutrition, is currently associate editor or member of the editorial boards of a number of nutritional journals. Professor Calder, the session is yours.
Great, thanks so much, Jeff. Hopefully you can see my presentation. So I'm going to kick off by talking about the immune system and how it works, and then some different aspects of the interaction of nutrition and nutritional state with immunity and how that might influence risk and severity of infectious disease, particularly viral infections. Just to start, this is my disclosures. So as I say, I'm going to start by saying something about the immune system, just to get everybody up to speed. Talk briefly about why different people have different immune competence, sources of variation in immunity. Then I'll mention the impact of obesity on immunity and susceptibility to infectious disease, make some points about nutrition in general, and then say something about 3 different micronutrients that I think are very interesting in the context of immunity, and then I'll summarize at the end. So the immune system is the way we protect ourselves from invading pathogens, things like bacteria, viruses, and so on. The immune system is composed of cells, but these often aggregate together in tissues like the spleen, lymph nodes, discrete pockets in the gut wall, and so on. But essentially, the immune system is our defense system. And we know that a well-functioning immune system provides good defense against pathogenic organisms. And we know that with certainty because people who have immunosuppression or who are immunocompromised are at much greater risk of infections and of infections becoming severe, even fatal. The 4 general functional features of the immune system are listed here. So firstly, it presents an exclusion barrier to keep pathogens out. So you could think of the skin, the mucosal linings of the gastrointestinal and respiratory tracts, and the acid pH of the stomach as being components of the exclusion barrier. Secondly, and really importantly, the immune system recognizes and identifies organisms if they break through the exclusion barrier. And this recognition is key to how the immune system protects us. Thirdly, it acts to eliminate those organisms that it recognizes as being harmful. So it actually destroys those organisms. It kills bacteria, it kills virally infected cells, so viruses have nowhere to replicate. Finally, the immune system has a memory component. So this is immunological memory. So your immune system can remember many of the encounters it's had before. And what that means, if a person is reinfected, the response is faster and more vigorous than it was the first time. And this memory component of the immune system is the basis of vaccination, which is very topical right now. So the immune system is pretty sophisticated with these recognition, elimination, and memory functions. And this high level of sophistication is brought about because of the many different components of the immune response. These are typically divided into innate or natural immunity and acquired or adaptive immunity. Innate immunity includes the barrier functions, but also some cellular functions like natural killer cells and other innate cells, and it includes the inflammatory response and the function of phagocytic cells, which engulf and destroy microorganisms. Acquired immunity includes the function of different T lymphocytes, and there are many different types of T cell, and B lymphocytes. Each of the different cells involved in the immune response has its own specific function. For example, B lymphocytes are the cells that produce antibodies. T lymphocytes are the controllers, and phagocytes carry out this process of engulfing microorganisms, for example. Some T cells, like cytotoxic T cells and also natural killer cells, they can kill virally infected cells and also tumor cells. So what we see is different cellular components that each have their own individual roles in the immune response. But all of these different cell types have to communicate with one another and interact to make an effective response to a particular organism. And I capture that in this figure from a review I published last year with regard to antiviral immunity. The detail of this doesn't matter, but what you see is the many different cell types are interacting with one another, ultimately aiming to defend us against invading viruses. So we see the communication between phagocytes, T cells, B cells, the production of antibodies, direct attack on virally infected cells by some immune cell types. So there's lots of interaction, there's lots of communication, and there has to be coordination. So again, just to summarize, the immune system presents a barrier to keep pathogens out, but it contains these really sophisticated cellular components of both innate and acquired immunity that come into play if the barrier is weakened or if these organisms break through the barrier. Of course, in the last 14 months, weak immune systems have been exposed as a major public health challenge, at least in parts of the world where there was less concern at the current time about infectious disease, as Geoff alluded to. So we've seen significant illness, significant mortality as a result of the presence in our environment of a new virus, which of course is highly virulent. Now we know some people show severe COVID-19, some people die as a result of that, but in fact probably most people who are infected show very mild symptoms. In fact, many will be asymptomatic. So some people are coping quite well with the presence of coronavirus, others are not. Why is that? Well, maybe it's because they have different levels of immunity, if you like, different immune competency. So here I show some of the many factors that can influence the immune response. And of course, these factors will vary between people. So there are some clear, obvious things like genetics will determine the immune response, infection and infection history, vaccination, some illnesses, some medications, things like cigarette smoking and alcohol consumption, they can impair the immune response. Stress, physical, physiological and psychological stress also impair immunity. But the immune response also changes with the life course. So you're going to hear later on about the immune decline that happens as people age, so-called immunosenescence. Physically fit people have stronger immune systems than unfit people. As I'm going to show you, body fatness impacts on immunity, frailty impacts on immunity. So overweight people and also people with frailty have impaired immunity. I'm going to talk about the influence of diet in general and some specific nutrients that support the immune response, and the gut microbiota, which again you're going to hear a little bit about later on, is also important in regulating the immune response. So there are many different factors involved that affect the immune response, and therefore it's unsurprising that some people in the population have stronger immunity than others. So I'm going to start by talking about the effect of body fatness. And I'm going to start right in with COVID-19, because relatively early on in the pandemic, it was noticed that there seemed to be a disproportionate number of people with obesity who were hospitalized with COVID-19 and who were displaying severe COVID-19 symptoms. So this is data from one of the first studies. These are French authors, where they simply related body mass index from normal weight to BMI more than 35 to the need for mechanical ventilation in patients who were already in the ICU with COVID-19. So these were all sick patients, but some of them required extra respiratory support. And what the authors identified was it's sort of a dose-response relationship, if you like, between body mass index and the need for ventilation. So in other words, the disease was more severe the higher the body mass index. Now, this may not be a surprise if you know about the literature on obesity and immunity, because it's very well described that obesity impairs the activity of many cells of the immune system, including cytotoxic T cells and natural killer cells that are involved in antiviral immunity. Obesity reduces antibody responses and reduces the production of an important antiviral cytokine, interferon gamma. It was already known that people with obesity are more susceptible to infections, and they don't respond so well to some vaccinations, including the seasonal flu vaccination, compared with normal weight people. In the influenza pandemic of about 10 years ago, people with obesity showed poor antiviral responses and recovered poorly from disease compared to normal weight people. Also, even when obese people are vaccinated with the seasonal flu vaccine, they have a higher risk of developing influenza compared to normal weight people. So the immune system is weak in people with obesity. This predisposes to poor vaccination responses and predisposes to infection. Paradoxically, obesity is associated with low-grade inflammation. So this is a real imbalance in the immune system, not enough immunity, too much inflammation. Now, this is a really interesting study where American researchers, this is Melinda Beck's group, took cells from healthy weight people or normal weight people, overweight people, and people with obesity, and they incubated those cells in a test tube with the influenza vaccine. So they were looking at the ability of cells from people of different body mass index to respond to vaccination in a test tube. And the outcomes were activation of cytotoxic T cells, important in antiviral immunity, the production of this protein, which is one of the proteins that helps the immune system kill virally infected cells, and the production of interferon gamma. And you see that the response was always weaker if the cells came from people with obesity compared to if the cells came from people of normal weight. And cells from overweight people were somewhere in between. So the immune cells of people with obesity have poor responses. Let's move on to talk about specific nutrients. So people often struggle with the idea that nutrition can be important in supporting the immune system. So here I list 7 reasons for nutrition being vital for the immune system to function. Firstly, the immune system has a high demand for energy. And of course, the fuels required for energy generation come from the macronutrients that we eat. Secondly, the immune system is highly biosynthetic. I mentioned the production of antibodies, cytokines. Also, there's huge cellular proliferation. So there's lots of biosynthesis going on. And of course, the building blocks for this biosynthesis come from things that we eat. Some nutrients are very important regulators of the molecular and cellular aspects of the immune response. Zinc, vitamin A, vitamin D would be good examples of that. Some nutrients are substrates for chemicals involved in immunity. For example, the amino acid arginine is the substrate for nitric oxide production, and nitric oxide kills bacteria. As I'll show you shortly, some nutrients have specific anti-infection roles. Again, zinc and vitamin D would be good examples of that, and vitamin A, and maybe selenium as well. Part of the immune response involves inflammation, and the aim of inflammation is to create a hostile environment for pathogens. That environment is also dangerous to our own cells, so we have to be protected from the oxidative stress and the inflammatory stress that the immune response imposes. And many antioxidant vitamins, minerals, other nutrients might be important there. And then finally, as I mentioned already, and as you'll hear later on, the microbiota is really important in supporting the immune response. And of course, our diet controls our microbiota. So one way to think about this is nutrient supply influences nutrient status in nutrient stores, and that influences immune function. And in turn, immune function determines our ability to defend against pathogens. So if we have an inadequate nutrient supply, too little energy, too little protein and energy, micronutrient deficiencies, we have inadequate status in stores, we have impaired immunity, and we have impaired ability to defend against pathogens. That means we have more infections, more severe infections, infectious illness, and death. So people have been interested in which nutrients are really important in supporting the immune response. And, researchers have been looking at this for decades now. Now, it's been identified, in fact, that there are multiple nutrients that are really vital for the immune system to function optimally. This includes fat-soluble vitamins, particularly A, D, and E, water-soluble vitamins, all of the B vitamins, vitamin C, many minerals, including zinc, copper, selenium, and iron, essential and other amino acids, essential and other fatty acids, and also, some of the polyphenolic compounds from plants all seem to have important roles. If you're interested in micronutrients, about a year ago, this review on micronutrients, immunity, infection, a very comprehensive review, was published in Nutrients. And this is a figure from that review, which hopefully you can see. So what the authors have got is the barrier function here. They've got the innate cellular components of the innate immune response here, the inflammatory response, antigen presentation, T-cell mediated immunity, and B-cell mediated immunity. And then these darker circles are the nutrients, the micronutrients that are really important for supporting that component of immunity. So within any of these circles, you see multiple micronutrients listed. So for T-cell mediated immunity, we see vitamins A, D, C, E, B6, B12, folate. We see zinc, iron, copper, and selenium. So multiple micronutrients are important for all these aspects of immunity. And in fact, the same micronutrients are listed in multiple boxes. So multiple micronutrients have multiple roles in supporting the immune response. So I'm going to talk about three of these in the context of COVID-19, vitamin D, zinc, and selenium. So first of all, vitamin D has many roles in the immune system. Immune cells have the vitamin D receptor, so that means they can respond to vitamin D. And interestingly, some cells of the immune system, including macrophages and dendritic cells, produce the active form of vitamin D, 1,25-dihydroxyvitamin D. Normally, we think of that being produced by the kidney. That's the sort of the classic pathway of synthesis of the active form of vitamin D. But in fact, as I say, dendritic cells and macrophages also produce the active form. And I think the fact that the immune system produces the active form of vitamin D tells me that vitamin D must be very important to immunity. Vitamin D plays roles in controlling the function of antigen-presenting cells, T cells, and B cells. It also promotes the release of antibacterial proteins like cathelicidin, beta-defensins, and overall it has important roles in both antibacterial and antiviral defenses. This is a meta-analysis of the effect of vitamin D deficiency on the response to seasonal flu vaccination. The seasonal flu vaccine includes 3 different strains of the flu virus, the H1N1, the H3N2, and the B strain. So you can measure in a person's blood antibodies to each of these 3 viral strains after a person has been vaccinated. And what the meta-analysis shows is that vitamin D deficiency impairs the response to the B strain and the H3N2 strain of the flu virus. So vitamin D deficiency impairs the vaccination response, so it impairs the immune response. That would relate to or translate into increased susceptibility to infection with low vitamin D status. So these are data from over 7,000 middle-aged British adults who gave blood in different parts of the year. Vitamin D was measured, that's the grey histogram on the left here. And so you see vitamin D levels are highest in summer, lowest in winter. And respiratory tract infections were monitored in those same individuals across the year. Respiratory tract infections are high in winter, low in summer. So this is an inverse association between vitamin D status and respiratory infections. Now, on the right is if you look within any season, winter, spring, summer, autumn, and you look at the effect of vitamin D status within a season on respiratory infection. And whether it's winter, spring, summer, or autumn, those individuals with highest vitamin D status always have the lowest risk of respiratory infection. Of course, this is just an association. It doesn't show cause and effect. Cause and effect would come from randomized controlled trials giving people vitamin D. In fact, quite a number of those trials have been done, vitamin D versus placebo, looking at respiratory tract infection. So this is a meta-analysis published in British Medical Journal in 2017 of 25 trials in 11,000 adults and children giving vitamin D with different dosing regimens and different doses, and obviously different durations, and then reporting on respiratory tract infections. And what the researchers found was that those individuals given vitamin D had a lower risk of respiratory tract infections than those individuals given placebo. So in other words, vitamin D prevents some, lowers the risk of respiratory tract infections. In subgroup analysis, they noted that patients who were vitamin D deficient gained most from vitamin D supplementation, which of course makes sense. So people have been quite interested in vitamin D in the context of COVID-19, and there are dozens of papers associating low vitamin D status with incidence of COVID-19 and severity of COVID-19. Of course, again, these are just associations. They have been subject to systematic reviews and to meta-analyses. So this is one of the systematic reviews that identified that studies showed that blood vitamin D status determines the risk of infection with COVID-19, seriousness of COVID-19, and mortality from COVID-19. This is a meta-analysis looking at vitamin D deficiency, showing that vitamin D deficiency increases the risk of severe COVID-19 compared to vitamin D sufficiency. Vitamin D deficiency increases the risk of hospitalization with COVID-19, and vitamin D deficiency increases the risk of death from COVID-19. Again, all from association studies, but quite convincing data. There are some randomized controlled trials of different robustness, looking at giving people vitamin D or not, and outcomes from COVID-19. Often these have been done in people once they reach hospital with COVID-19, giving them normal care with and without usually high doses of vitamin D over the first few days of hospitalization. In fact, these trials have already been subject to meta-analysis, and this is the effect of vitamin D supplementation on length of ICU stay. And you see vitamin D supplementation reduced length of ICU stay by about 90% in the meta-analysis of these three studies. So vitamin D supplementation in people hospitalized with COVID-19 seems to improve outcome. Let's look at zinc now. Zinc also supports the function of many different cell types, contributes to antioxidant protection, reduces inflammation, and as I'll show you in the next slides, it has very specific antiviral actions. Supplementation studies with zinc, some older studies, some newer studies, show that giving people with low zinc status or low zinc intakes, these are usually older people in these trials, giving them supplemental zinc has been demonstrated to increase, to improve some immune biomarkers like T cell function, for example. There are also some studies showing zinc improves response to vaccination, and there are quite a few trials looking at the ability of zinc to treat infectious illness, both diarrheal and respiratory infections. And indeed, there are meta-analyses showing that zinc can be used to treat severe pneumonia. So zinc seems to be important in supporting the immune system, preventing viral infection, and maybe even treating viral infection. Now, one of the intriguing things about zinc is it has specific antiviral actions. So this is coronavirus. The genome of coronavirus is single-stranded RNA. For the virus to replicate, it has to produce another copy, a new copy of its genome. It has to produce viral protein, and then the virus subunits are assembled. Now, the first step in both the RNA synthetic pathway and the protein synthetic pathway is catalyzed by this enzyme, RNA-dependent RNA polymerase. And it turns out that zinc is an inhibitor of RNA-dependent RNA polymerase. So zinc may have a specific role in preventing replication of coronavirus. Now, this is nicely shown in this paper from 11 years ago by Dutch researchers, where they did cell culture studies looking at the effect of zinc on coronavirus replication. So they infected epithelial cells, and remember, coronavirus infects respiratory epithelial cells amongst other cells. They infected epithelial cells in a laboratory with coronavirus, and then these are the different coronavirus RNAs that are produced, and then they added to the culture medium increasing concentrations of zinc. And you see, as the concentration increases, so the amount of viral RNA decreases. So, of course, this wasn't the only experiment they did, but their experiment showed quite clearly that zinc can prevent coronavirus replication. Now, the concentration of zinc in the bloodstream is around about here. So this is, we may be able to get additional antiviral effects by raising the concentration of zinc in the bloodstream or in interstitial fluids. Now, just coincidentally, about 18 months ago, there was this really nice review on zinc and antiviral immunity and advances of nutrition that talks about the different parts of viral biology that zinc interferes with. So that's pretty interesting. Now, again, people have been interested in zinc and COVID, of course. So these are the sorts of studies that have been produced so far. This is a study looking at zinc status in healthy controls and people hospitalized with COVID-19. People hospitalized with COVID-19 have lower zinc status than healthy controls. Those who are discharged from hospital had better zinc status than those who died as a result of COVID-19. Another study showing lower zinc status in patients with COVID-19 compared to healthy controls, and another one showing zinc is lower with severe COVID-19 compared to mild or moderate COVID-19. And then this is a receiver operator characteristics plot where the authors identified that low zinc was a very highly sensitive and specific marker or predictor of critical illness in COVID-19. There are a couple of randomized controlled trials of zinc in patients hospitalized with COVID-19 showing that zinc can improve outcomes and reduce mortality. The last nutrient to talk about is selenium, which I think is much overlooked. So selenium supports the function of many cell types, contributes to antioxidant protection, reduces inflammation. It improves immune biomarkers, at least in some studies, and it has been used in various viral infections with some benefits. Now, there's been quite a lot of research on selenium deficiency in mice, and these studies show that selenium deficiency impairs the immune response, increases susceptibility to viral infection. Selenium deficiency allows viruses to mutate, including the flu virus, and it allows normally weak viruses to become more virulent. And I think this is really interesting in the context of the emergence of coronavirus variants, whether these emerge because some people have a nutritional environment, physiological nutritional environment that permits viral mutation. I think that's an interesting question. Now, for me, the key study of selenium and immunity is this study carried out in Liverpool, published in 2004. So this is a study in adults with low selenium status who were put into a randomized controlled trial, and they received placebo, 50 or 100 micrograms of selenium per day for a period of time. And then they were given the oral poliovirus vaccine. So the poliovirus vaccine given orally, it's not injected, and it's a live poliovirus. It's live but attenuated. So it's still alive, but it's not dangerous. Then they took blood from these people and they stimulated the blood in a test tube with the poliovirus. So before vaccination, this is interferon gamma production. Before vaccination, these cells from these people don't respond to poliovirus. Post-vaccination, they do. They produce interferon gamma. So this is interferon gamma at 7, 14, 21 days in the placebo group in the dark bars. But you see interferon gamma production is increased at 50 micrograms of selenium per day, and it's increased even more at 100 micrograms of selenium per day. So giving selenium seems to improve T-cell responses and interferon gamma production in response to poliovirus exposure. Now, because the poliovirus is alive and is given orally, if it's not eliminated from the body, if it's not destroyed, if it's not contained, the virus appears in the feces. So this is the number of people, 20 per group, who had poliovirus in their feces at 1, 2, and 3 weeks post-vaccination. So in the control group, even 3 weeks post-vaccination, 14 out of 20 individuals had poliovirus in their feces. This was lower, it was 9 in the 50 micrograms per selenium per day, and even lower, 7 out of 20 at 100 micrograms of selenium per day. So selenium results in better viral clearance. The other thing these researchers looked at was mutant viral sequences in the feces. So on the left of the bottom panel, these are fecal samples from the controls, 50 micrograms of selenium in the middle, and 100 micrograms of selenium on the right-hand side. This is the normal poliovirus sequences here. In the control group, you see the emergence of these mutant sequences. So even over the course of a few days to weeks, the poliovirus is mutating in the individuals in the control group. This seems to be less at 50 micrograms of selenium per day, and it seems to be almost absent at 100 micrograms of selenium per day. So giving supplemental selenium prevents viral mutations in humans. Or to put it the other way around, selenium deficiency permits viral mutations to occur in humans, just as it does in mice. So again, people have been interested in selenium status in COVID-19. Again, these are data showing selenium status is lower in people hospitalized with COVID-19 compared to healthy controls. It's higher in those who are discharged compared to those who died. So to summarize, what I've told you is the immune system is central to protection against infection. It's highly complicated, it's highly sophisticated. I mentioned that obesity is associated with impaired immunity, so is frailty. So undernutrition and overnutrition both impair immunity. I've talked about the role of specific nutrients and particular vitamins and minerals that have important roles in supporting the immune system. I've told you that low intakes and status of these nutrients impairs the immune response and makes people more susceptible to infections, including viral infections, and this situation can be prevented or reversed by repletion. Zinc, and maybe vitamin D as well, has special roles in antiviral immunity, and I think this effect of selenium, which no one is really talking about very much, is really intriguing. So I think to really wrap it up, nutrition has a key role in dealing with pathogens, both bacteria and viruses, and in preventing infection becoming more severe, and micronutrients are really vital for this. Thanks very much for your attention.
Fantastic. Thank you very much, Professor Calder. Excellent introduction. So much depth of information, really interesting. So I think we'll leave the questions to the end. And now I'll return the chair over to Professor Calder to introduce the next speaker.
Yeah, great. Thanks so much, Jeff. So it's a huge honor for me to introduce to you Professor Tamas Fülöp. So Tamas is from University of Sherbrooke in Canada, and he is one of the global leaders in studying the effect of aging on the immune system, this process of immunosenescence. So it's really wonderful that we can have Tamas here with us today to talk about this really important feature of aging. Tamas, over to you.
Thank you very much for this very kind introduction, and also thank you for ILSI and Professor Calder for inviting me and giving me the opportunity to speak this morning. So, of course, this is a huge comment to speak on aging and immunity, but I will try to make a sort of overview of what's going on in immunity and aging. And thank you, Professor Calder, for setting the stage to explain how immunity works, so it will make my task easier. So the next slide, please. So this is my disclosure. The next slide, please. These are the abbreviations. So if you get the slides, so you can refer to that. Next slide, please. So aging is meaning time. So you can see here the lady who was the most aged, oldest person in the world, who is Jan Calmet, who was about 122, which is considered actually is the maximum lifespan of human beings. And you can see that once she was young, middle-aged, and elderly. So when we are speaking on older subjects, we should really consider, as already mentioned by Professor Calder, that the immunological history, what we can call actually as after Professor Franceschi, the immunobiography is very important. Of course, as we have seen, many other factors are influencing the immune system, but this is one of the most important when we are looking on the effect of aging to the immune system. The next slide, please. The next slide, please. So I wanted to make the stage that immune system is part of a huge other system, which is aging. Aging is a very complex system determined by external and internal factors, and they are interacting at different levels from the molecular to the organism level. So there are interactions, there are feedback, there are many controls which are in movement when you consider what is aging. And the immune system is part of this very complex system and determined by every other systems which are present and determine how we will age. The next slide, please. There was a very big step forward when the group of Lopez Otin, and some years ago, determined the 9 hallmarks of aging. And this was a sort of conceptualization to make really evident what is the main processes inside of our organism which can define how aging is occurring with time. So you can see many of that, and you have, of course, the deregulated nutrient sensing. So as we have heard before, so it's very important, but there are many others like for the metabolism, the cellular senescence, the stem cell exhaustion, loss of proteostasis, epigenetic alterations, telomere attrition, genomic instability, and finally, what we call the altered intercellular communication. Next slide, please. So inside of this 9 hallmarks and inside of this intercellular communication, you can see beside the neuroendocrine dysfunction and bystander effects, 2 words, which is immunosenescence and inflammaging. And so you can understand that immunity and the part of the immune changes with aging are part of the aging process and from the 9 hallmarks of aging. Next slide, please. So this is from 2013 when the group of Goronzi just summarized very clearly what's going on in the immune system with aging. And also he contributed to the understanding of the adaptive part of the immune system, but things are progressing and our thinking is evaluating. And you can see here that this was, and this is still sometimes the concept that what we see in aging in the immune system is a sort of immune deficiency. So I will try also to show you that beside having some decrease in the immune system, we have also other processes which can make our immune system stronger when we age. And of course, you have the inflammatory processes as we age much higher in case of older subjects. The next slide, please. So what is immunosenescence? Next slide, please. So as we have heard, the immune system has two major parts, which is the innate immune system, which is a sort of standard for every multicellular living organism, and we have the adaptive immune system, which started very late in the evolution of the beings and the animals on the earth. So the innate immune system is a very ancient, which can react with almost everything that we have outside of our organism and also inside of our organism. So this is reacting to the pattern molecular the conserved pattern and also to the damage associated molecular patterns. So every viruses, bacteria or fungi that we have can react and find when they are in organism the innate immune system. You can see here they really call an inflammation that this is to eliminate invader. And in most cases, this innate immune system sufficient to the invaders, but it is not. The system is also its part to the of the aggression. So this is composed of cells, neutrophils, macrophages, cells, NK cells can really by various outside or inside cells and can have very produced. And here that specific stimulation of the major functions of these cells are decreased with aging, namely the chemotaxis, phagocytosis, the intracellular killing of bacteria or even viruses, and also the production of various mediators which can eliminate outside of these cells these aggressions. The next slide, please. So the next slide, please. So the paradox of the aging of the innate immune system, paradox of the pattern recognition receptors, that in the come back, please. Sorry. So this is a constitutive. That's it. The constitutive activation of this system at the quiescent state. So which means that these cells and the increase of the mediators will sign a sort of hyperactivation state with time in elderly or older subjects. But when it's time to react clearly to a specific aggression, the specific activation is decreased. This is not meant that older subjects cannot fight infections, but this means that efficiency of this may be decreased. And this is because we have all these external and internal stimulations constantly, and this maintain the readiness or the alertness of the innate system with aging better at the quiescent state than when they meet specific aggressions. The next slide, please. This is to show up at the red arrow that in case of elderly subjects, already many signaling molecules inside of the neutrophils are in an activated, which means phosphorylated state. And when you try to stimulate specifically these cells, you have a decrease in this phosphorylation. This was our work, which was published some years ago. And the next slide will show the same from the group of Professor Janet Lord from Birmingham, where they show exactly the same. Then once you have the basic activation of these cells, which impact the functioning of these cells, you cannot further stimulate these cells. So this is a very important observation for the innate immune system with aging, the higher activation at the quiescent state and the less activation during activation and specific stimulation. The next slide, please. The one other very important task and function of the innate immune system, as already mentioned also by Professor Calder, is the antigen presentation. This is absolutely fundamental for a good functioning of the adaptive immune system. This is just to show you that we can have presentation by mature dendritic cells, by macrophages, by follicular dendritic cells, and these have a very specific task towards the specification and the activation of the adaptive immune system through the CD4 T cells, CD8 T cells, and the B cells. And their counterpart in this are the naive B naive T cells, CD4 and CD8 cells. And when we have problems in this antigen presentation, as we have with aging, which means that the processing of the antigens is decreased, and also the presentation through the MHC2 or the MHC1 epitopes for these antigens is decreasing, there are problems for priming the adaptive immune system to work properly after an infection or an aggression. The next slide, please. So there are changes in the adaptive part of the immune system. There are phenotypic changes and there are functional changes. We'll see first the phenotypic changes, which are very much studied by thousands of publications and many groups. And what is the consensus? That there is decrease of the naive T cells with the loss of their possibility to have diversity at the TCR level, and there is in the same time an increase in TCR oligoclonality, which are meant to one antigen, and also a very big increase in the effector memory T cells with a loss of the CD28 on their surface. So the next slide, please. So you can see here at the cellular level, there are changes in the B cell compartment on the left, and you have in the T cell compartment on the right, the same changes, that the decrease with age of the naive B and T cells, the increase in the memory T cells, and also the increase inside of these memory T cells of the exhausted and senescent T cells, as we will see later. And in the case of B cells, we have also an alteration in the immunoglobulin specificity in the isotype and at the idiotype level. So the immunoglobulin switch to perfect and adapted immunoglobulin IgG production is altered with aging. The next slide, please. So there are many questions actually still to determine what is the role of the cytomegalovirus. Many persons think that the cytomegalovirus is one of the initiator and maintenance for these changes with aging. but most of the time we know that there are changes which are caused by CMV latent infection with the reactivation time to time and also with the aging process. So actually we know that CMV may contribute in some individuals, but most of the time the aging of the immune system is independent from the CMV infection. The next slide, please. So there are four groups of T cells, which are the naive T cells, the central memory T cells, which keep some of the aspect of the naive T cells, the effector memory T cells, and terminally differentiated memory T cells, re-expressing the RA phenotype. So you can see here that with aging, we have a shift from the naive T cells towards the effector memory and the TEMRA cells, which has very specific phenotypic expression and corresponding to different functionality. The next slide, please. So when we see how a naive T cells can evaluate when they are stimulated with a specific antigen and coming memory T cells, you can see here that with aging we have an increase in the TEMRA, which can express some NK features, so they become more innate than adaptive. You have what we call the exhausted T cells, as we will see later, and also we have a short-lived effector T cells, which are increased. But in the meantime, the stem-like memory T cells, which keep some of the possibility to divide as a naive T cells, are decreased, and also the functional tissue-resident memory T cells are decreased, which means that we have many cells which can, have effector functions, but these effector functions are less efficient than in the case of a younger subject, because all the network of these memory T cells are changing in their composition. The next slide, please. So when we consider the functionality, the most important functionality is the oligoclonal proliferation, which is decreased with aging. This is because the IL-2 secretion is decreased, but also because the CD28 receptor at the surface of the T cells is decreasing. In the meantime, you have the decrease in the telomere length, and the telomerase induction is also decreased, and concomitantly the damage to the DNA is increased. The next slide, please. So you can see here very schematically the differences between the inducing of the functionality of these T cells through the membrane and the receptors on the surface of the T cell, and also the intracellular pathways, signaling pathways, which are altered with aging. So in the young, we have a feed-forward mechanism, which is much higher than the feed backwards. But in the elderly, unfortunately, these cells are under inhibitory control, which means that even if they are stimulated, they have phosphatases, they have blunting in their different intracellular signaling molecules, which make them less responsive to specific antigens which are coming from outside or inside and presented by the antigen presenting cells. And you can see here that there are changes in the AKT-mTOR pathway, which is very important for the immunometabolism and also for the maintenance of the balance between the metabolism needed as aerobic glycolysis or the OXPHOS metabolism during the stimulation and the differentiation of these T cells under antigen presentation. And you can see also here the changes in the chromatin and also the epigenetic changes that we will discuss later. The next slide, please. So you can see here, as it was presented and shown for the innate immune system, the quiescent hyperactivation in the quiescent state. You have the same from the Shen-Or group, which was presented also in T cells, that namely the state STAT molecules were also, which are the intracellular pathway for cytokines to transmit the signals from the outside to the nucleus. They are also in an activated state in T cells of older subjects. The next slide, please. So as you can see here very briefly, that the epigenetic changes mainly at the histone methylation. The histone changes by methylation, acetylation. So the histone modification and also the DNA methylation will really determine which part and how the DNA will be transcribed. These are transcriptional regulator of the DNA. So the next slide, please. We can see that beside all the changes that we have at the level of the DNA transcription and the epigenome modification and control, we have a global hypomethylation, and also the heterochromatin, which is less transcribed, is lost, and there is a redistribution. So the restriction and the inhibition is less, and the activation is much higher with aging in these T cells through the development of naive T cells towards the differentiated and very differentiated states of memory T cells. The next slide, please. So one of the major underlying cause of these changes, beside the post-translational changes, is the involution of the thymus. The thymus is starting to involute very early after the puberty and almost not existing at the 60 when we attain this age. And this makes changes in the production and the priming of the T cells. So, which you can see on the right side, that the naive T cells are decreased with aging, because there is changes in the epithelial and in the hormonal level in the thymus. But in the meantime, you have an increase in the self-reactive T cells and also in Tregs, which are really increasing the possibility of autoimmune diseases and also a negative control on these self-reactive T cells. So this is leading to what we call the inflammaging. The next slide, please. So the conclusion from this first part is that elderly are immunoremodeled with fewer naive T cells and dysfunctional T cells due to chronic antigenic stress and the thymic involution, which is coupled with innate immune response, resulting what we call the inflammaging. Next slide, please. So we will look what is inflammaging and what are the sources which led to that. So we already saw that one of the main reason of this inflammaging is the disbalance between the innate and the adaptive part of the aging of the immune system, when the hyperactivation of the innate immune system outcompete the changes what occurs in the adaptive part of the immune system. And this was the base. The next slide, please. of the conception by Franceschi at 2000 of this aging concept, because of this chronic antigenic stress and oxidative stress which arise each day in our organism and accentuated by the aging of the organism and the stimulation by tumor antigens, beta amyloid, oxidized LDL, age products and others, stimulation by the persistent viral infections that we have through our life with the HSV1, with the varicella zoster, the CMV, which leads to the changes that I have shown you before. The next slide, please. So this is interesting that the inflammaging and even aging is an evolutionary unpredicted byproduct of the degeneracy of damaged sensors, which means that with aging we have more and more sense for the molecules which are stimulating our system, and there are really a limited number of evolutionary selected promiscuous sensors to all these numerous stressors, and which is mainly are on the macrophages, which made that the inflammaging is really macrophage centered. But as you can see, the inflammaging and the fan out of this huge number of stressors and less sensors is even less molecules which can be the effector of this inflammaging, namely pro-inflammatory cytokines. The next slide, please. So one of the inner stimulant of this process is what we call the circulating mitochondrial DNA. Not only the circulating, but also the intracellular mitochondrial DNA, as we know, actually can be one of the powerful stimulant of this inflammaging. So we don't need always from outside infections or other triggers, but from the inside, we have already quite a lot of changes which can fuel what we call inflammaging. The next slide, please. So you can see here how the mitochondrial DNA can lead through the inside TLR receptors, which are the TLR3, 7, 9, to the activation of the NF-kappa B inflammatory pathway, and also by stimulating what we call the inflammasome and resulting in increased pro-inflammatory cytokines. The next slide, please. It should be also mentioned that this mitochondrial DNA is also very important to stimulating what we call the mitochondrial antiviral signaling protein, which is really important to stimulate and put into movement the intracellular pathways to produce two very important antiviral cytokine, which are the type 1 interferon, which are stimulated also in the meantime by the mitochondrial DNA through the cGAS STING pathway, which is very important for the cellular defense against viruses. The next slide, please. So we have seen the nucleic acid, how it can fuel the inflammaging. We'll go to the two other parts, which is cellular senescence and the gut microbiota, very briefly. The next slide, please. So one of the major advance in aging and also in immunity in aging since the Hayflick theory, which was the replicative senescence, that we could really sense how these senescent cells can increase with aging and also fill the tissues, and even if they are not capable to divide, they are still there, and they are metabolically extremely active because they can be stimulated via these different pattern recognition receptors, and they become what we call the senescence associated secretory phenotype. And this means that these cells which attain a certain limit of their proliferation capacity become resistant to apoptosis and fill the tissues and the space and fuel the inflammaging by their inflammatory products. The next slide, please. And these cells are should be distinguished, and this is a big debate in the immunity on aging, whether the T cell exhaustion is the same than the T cell senescence, what are really the proportion of these cells, and this is extremely important when we think about the treatment of diseases and the treatment capacity that we have with the checkpoint inhibitors, that the T cell exhaustion can be reverted and they become functional, but the T cell, the senescent T cells are not able to be activated or be really useful for the organism, and they should be eliminated. So the next slide, please. So this is to show that, and we will hear on that, and was also mentioned, that good microbiota, eubiosis, is very important for the efficient and really good working immune system. This is just to show you how the interaction between the barrier and the cells and the different component of the microbiota can direct and influence the immune system. The next slide. With aging, of course, we have many changes in the microbiota, which doesn't mean that it's pathological, because there are increase in some type of component species of the bacteria which compose the microbiota, and also the decrease, the increase. So there are various changes, but nevertheless, these changes can influence the changes in the whole immune system with aging. What is interesting, the next slide please, that the microbiota in very old, in the extreme longevity, in the semi-supercentenarians, is completely different from elderly subjects. So we know that in the blue zones where there is an increase of these extreme longevity persons, the semi-supercentenarians, they have a completely different microbiota, even from young or elderly, between 65 and 75. And we know that even they are different, they are more close to what the young persons can present than the elderly, which means that many factors that we have heard in nutrition and lifestyle can influence, and with the genetic background can maintain a microbiota which can sustain a normal immune response. The next slide, please. So this is to show how the different nutrients and diet can influence how the microbiota can have the modulation of the immune system, and the Mediterranean diet is mainly maintaining immune tolerance and a good immune functioning, and mostly the Western diet with their components may lead to what we call the inflammation and also influence the inflammaging. The next slide, please. So altogether, the senescent cell hyperfunction and also the innate immune hyperfunction are leading to organ damage, age-related diseases, and finally death. I will just mention briefly one or two diseases where this is important. Next slide, please. We cannot, we have heard also on COVID-19, and we know that impact more in the severity and also for death, the elderly subject, which is shown here. The next slide, please. And many things with the aging process inside of the immune system can influence the susceptibility of elderly subjects to the COVID-19. And you can see here the uncontrolled immune response, the epigenetic dysregulation, immunosenescence, inflamed aging, the biological age, which is higher than the chronological age, and of course, environmental factors and comorbidities will influence the susceptibility. The next slide, please. This is a busy slide, but just to mention that the decrease in the naive T cells with aging that you can see on the right side, even in CD4 or CD8 T cells, may highly influence the response and the susceptibility of elderly subjects toward COVID-19. The next slide. And not only the decrease in the naive T cells, but the uncoordination which is occurring with aging between the different part of the adaptive immune system, namely the production of neutralizing antibodies against the S protein and also the effector functions of the CD4 and CD8 T cells as the memory formation should be made in a real coordinated way towards a viral infection, and even more importantly, towards the COVID-19, the SARS-CoV-2 infection, which means that in the lower part of the slide, you can see that there is a complete uncoordination between the production of antibodies and the function of the T cells. The next slide. Why it is so? Also because there is a preponderance of the macrophages and monocytes in old subjects infected with SARS-CoV-2. And you can see where you have the arrow that compared to young healthy and aged healthy subjects, aged people who has the COVID has a higher monocyte activation and higher monocyte number. And also when you compare young COVID and aged COVID patients, you can see that there is an imbalance between the cellular compartment of the immune system, and we have a hyperactivated immune state that you can see on the bottom part of the slide with the expression of the genes which are related to the hyperactivation inside of the innate system. So which means that one of the most important susceptibility to COVID-19 in the elderly is the hyperactivation of the innate immune system, which could exist before the infection by this virus. The next slide, please. So these are all the components which can determine why elderly has a higher susceptibility towards the COVID-19. The next slide, please. So one very important disease of the elderly is Alzheimer's disease. And you can see, and nowadays this is well accepted, and it took perhaps 30 years to develop and to link the Alzheimer's disease to the neuroinflammation. And this is a neuroinflammation which makes, after the lifelong stimulation of the neuroprotective microglia to neurotoxic microglia, and being part of what we call the inflammaging process, and destroying at the end of the road the cholinergic neurons and leading to these very important neurodegenerative diseases. So it is a very quick overview, but this is part of what I told you on the changes in the immune system with aging. The next slide. This is to show you that about 30 years before the clinical manifestations of the Alzheimer's disease, you have all this chronic antigenic stimulation, either by beta amyloid or by infections, which leads to what we call now the neuroinflammation. And once the aging of the microglia is manifest, you can have the clinical manifestations of the Alzheimer's disease. The next slide, please. This is to show that the same is happening in cancer with all the outside, inside stimulant stressors during the lifetime of the individuals. The next slide, please. So this is to mention that during these last years, we have what we call the innate immunity, which has now what we call the trained innate memory, which means at each time that you stimulate the immune system and the innate immune system especially, there are changes in the immunometabolism and in the epigenome control, which makes these cells more sensitive. And when you have following and repetitive infections or aggressions, you can have a higher response. So this is a non-specific memory, but this makes the organism much more ready to answer to a second, a third challenge that was the first one. Of course, we should mention that this is not only a positive way to control infections, but can be also, as we see sometimes in aging, could be a lead to what we call the immune paralysis and not response anymore, and making more inflammatory molecules for destruction inside of the organism than the destruction of the aggressors. The next slide, please. This is to show you how the epigenetic changes inside of the monocyte macrophages and NK cells possessing that trained innate immunity can control finally the higher response when a new aggression is occurring inside of the organism. So we think that the inflammaging is a sort of mirror of what's going on in the trained innate immunity at the innate immune level. The next slide, please. So this is to show you that after a hip fracture, there are still production of TNF alpha. Okay, shortly I will finish. Then, yes, the next slide, please. This is the geroscience, and the next slide, please. Geroscience is stating that the inflammaging is incorporated more clearly inside of the 9 hallmarks of the aging process. And this means that, the next slide, please, all the different processes inside of the aging and including the inflammaging are the common root for all the age-related diseases. So if we can really impact these processes, the aging will, the diseases related to aging will be mitigated. The next slide, please. There are, they don't really take into account that the inflammaging is balanced by the anti-inflammaging. And this means that at the same time when we have an inflammation, we have also anti-inflammatory control. And you can see that the balance between inflammation and the anti-inflammation can lead either to age-related diseases or frailty, or to successful aging or longevity. The next slide, please. So this is conceptualized in this way of aging, that there are 3 types of aging, which is the one is the pathological, the normal, or the successful. And this is mirrored by what we can see inside of the immune system. The next slide, please. So this is the comparison of the 3 types of immune trajectories which are leading and communicating with each other. The geroscience straight link from aging to the diseases, the Franceschi conceptualization with the differential trajectories which are modulated by the outside and inside factors, and the interrelation between the aging and the age-related diseases. diseases. The next slide. The next slide, please. So this is the new paradigm of the dynamic immune changes with aging, that the immune changes are not all detrimental, but these are part of the adaptive immunity that make the older organisms sometimes more efficient, but the adaptation can be maladaptation when we have all these different diseases. The next slide, please. The next slide. Next slide. This is the summary and the perspectives. So the immunosenescence should be considered as a remodeling adaptation of the innate and adaptive immune system in function of chronic aggressions and time. It is necessary process of adaptation, but detrimental for responses to new, for example, cancer antigens or viral antigens. The intricate relationship between immunosenescence and inflammation induces a vicious cycle of self-maintenance, which may result in maladaptation. Elderly in the clinical setting are doing much better than conceptualized from experiments. Real deep human longitudinal studies are needed, and the anti-inflammaging process targets the changing immune system of the elderly to contain inflammaging at an acceptable level. Thank you for your attention.
Great. Thank you very much, Tamas. This was fascinating insight into some of the paradoxes of aging and its impact on the immune system. So our final speaker of the session is Paul Ziemnisky. Paul is from Dairy Management Inc., and he's going to tell us about COVID-19 and the food supply chain. Thank you very much, Paul.
Good morning, everybody. Two things, I guess. After watching those fantastic presentations, hope everybody had their yogurt and milk today. You know, I think it's just showing we're at the tip of the iceberg, really, of it, understanding how food and what we consume can affect our bodies. And I think about the next 25 years where we're going to go linking food and science. From our side, on behalf of dairy management, I work for the dairy farmers, and that's where we're investing a lot in this future space. You talk about lactoferrin, we're just starting to understand things like that. My role, though, unlike my three predecessors, my background is brand marketing and innovation, and I was asked to talk about the COVID-19 and the food supply disruption. So what I want to do is just hit some highlights of what happened in the U.S. with some storytelling here, and then just get to let you guys get to questions. And so from, I'll call it about March 17th was D-Day last year when, the U.S. shut down. And we call it 4 weeks of chaos that occurred in the U.S. supply chain. And I'm going to focus really on the dairy elements of the supply chain. Dairy Farmers of America is the biggest co-op here in the U.S. And when COVID hit, you had mass chaos within the food industry and I'll talk about, I wanted to lead with the impact, and I'll talk about the whys, but they were dumping about 3.7 million gallons of milk back on the farm as we got the supply chain reorganized to handle the disruption. What we did as an industry, we formed what we call these emergency action teams to focus on how do we make sure that the kids are getting the right nutrition, since the schools shut down and people were getting educated at home. The hunger channel, we had a massive increase in food insecurity with people not working. And then the disruptions of food service where you had shut down, to give an example, you think about dine-in restaurants where 35 to 100% of their businesses is relied on dine-in, shut down. Where does all that volume of products that go through that channel? And then vice versa, you had retail and grocery, had 50 to 75% increase in consumption. So we'll talk about the chain and reaction to all this complexity and how the industry rapidly got together to figure out how they moved different packaging products around to meet these needs and changes. And I always like to think, sometimes we take advantage or take for granted the food system and how products move through the food system. So I just grabbed some slides to talk about that. When you think about from ingredient, it moves, milk comes in a tanker, it goes to whether it's a manufacturer or a distributor, and gets processed, and then whether it goes to retail, hospitals, universities, and the distributors that can handle that market, hotels, to the restaurant industry, whether it's a quick serve, fast casual, or fine dining, and then these smaller individual local places. So you've got a lot of products moving different sizes and packaging, and you've got emerging technologies out there, whether it's like the meal kits or I'll call it delivery companies that are popping up to get the food to the consumer. So, and that created, when COVID hit, packaging, what I called conundrums, where you have immediate oversupply because of the changing of the consumer environment, and then also immediate scarcity. So put that in perspective. When you think about schools, in the US, we have 1 billion packages and cartons that go through the school environment. Again, all these kids were not in school anymore, they're at home. And vice versa, you had people who weren't working again, you had massive growth in at-home consumption. And so, from a 50 to 75% spike, people weren't planning changes in their supply chain here. When you look at food service and sour cream as an example, you have 5-gallon tubs that are designed for distribution to food service. Retail, you had massive runs on your normal 8 to 12 to 16 ounce type of products. So what do you do with all this excess tubs of sour cream that have 30-day supply, 21-day supply, or on the shelf, when they're not sellable marked, that they're not, I'll call it FDA approved to sell at retail, and people aren't buying 5 pounds at the retails for their home use. So just a lot of challenges there in the food system. So then third, you think about cheese. You've got cheese designed for food service, backroom preparation, whether it's Italian, Mexican channel, different types of food service entities. What do you do with all that? And then retail, you have, again, a 50 to 75% run up a product. And then you think about the complexity of cheese, you also have a lot of custom packaged cheese. When you think about aged cheddar 90 days, you get a 75% lift. You just can't add 90 days of aging to meet that. And so you also run into supply and production issues pretty quickly due to the aging process. And then over the decades, there's been a lot of for mass efficiency packaging systems designed for optimization across channels. I just grabbed milk. Whether it's a restaurant, the C-store, or healthcare, et cetera, you've got all these, it's customized over the time, and the same thing with other types of packaging. So you've got this packaging effect of increased demand in some channels and dramatically declines in demands in others. And when you think about fluid milk, prior to COVID, just put it in perspective, in the US, 5 billion gallons, what is that about? 3 billion liters, something like that. And 75% had already gone through retail, but you had schools, which is 8%, restaurants, another 8%, other types of food service like hotels make, that makes up a quarter. So you had one section of the world seeing a dramatic increase and others, dramatic declines. And then I thought this was when you think about milk, so you go through, overall milk was flat, in the US last year. But then if you're trying to manage the packaging, you're trying to manage the channels and package size, and you retail, again, over 600 million gallon increase. And then schools, a 400 million gallon decline. And then all others, again, also declined. And so, just a dramatic pressure on supply chains, giving a very short shelf life product. How do you manage through that? So there's a lot of complexity. Like we don't want to dump it. So like I said, the first 4 weeks was chaos, but over time we worked across the industry to say, how do we take the school demand and meet with new types of package systems for retail, and also incorporate where that increased demand is in hunger. I'm just going to give you some highlights when you start to then break down the channels. When you look at COVID's impact to restaurants, both global and in the US, they almost mirrored each other. So the top bucket is, the restaurant sales demand at, from Australia to Mexico to the UK, everything was pretty consistent. You just saw this almost at dining in restaurants immediately drop off. So it put a lot of that restaurant-focused packaging that is not labeled for retail sale, and all those products with short shelf lives out there in a quagmire. And then, you know, the bottom is the US, and just again showed this drop where we've seen, and we still haven't completely recovered in the US and globally. And then, you know, from a big picture food service, I mean, you've had decades of decline of growth and sales, and all of a sudden you see again just an extraordinary decline of volume. And so, you know, the job impacts that it impacted in the US, supply, and again just chaos in the food industry chain. And then you have, I call, winners and losers. And when you start to fragment the category, you know, the winners in food at restaurants were set for digital. You know, like when you look at players like a Domino's, who've got a dramatic application system, you know, on their phone, that whether it's set up for delivery, set up for carry out, or like a, you know, like a McDonald's or the Paneras of the world, or like a Chick-fil-A here in the US, where they're set up for delivery, drive-through. They succeeded and maintained growth. But if you look at these casual and fine dining, where they have, you know, the individual proprietor hasn't invested as much into delivery systems and carry out systems and I'll call it new digital technology, they just were crushed. So then if you're a dairy manufacturer in the US that were, you know, doing fresh mozzarella for a fine dining Italian restaurant, you had and you just produced 30 days of supply, and your restaurant industry shut down. You know, that was a big quagmire here in the US. You know, shifting to the retail, and this is where you saw the opposite. When you look at like this is a fluid milk at retail, you know, we were averaging about 5% decline, and what's happening with fluid milk is, you know, really, you know, 30% of the declines are tied to cereal consumption. Cereal's declining because people are eating away from home, eating high protein bowls, etc., or bars. Other things, just competitive beverages. You know, so you're drinking it in your coffee and your creamers. So that was the historic declines, and it's easy to forecast and plan. When COVID hit, everybody's at home again. They're eating cereal again. They're actually baking more, you know, and drinking more at home. And those kids who were in school, who were, you know, getting the little cartons, were at home. They're starting to drink gallons. And so this is just, you can see the immediate spikes for the first, you know, 2 to 4 weeks. And then, as I talked about the inverse relationship with milk and cereal, I overlaid the graph of the cold cereal demand to the fluid milk demand, and you can see a big spike, and, you know, it's linear, of consumers at home, the rebuying and reutilization of cold cereal, and what it's done for fluid milk during the COVID period. And then, you know, this is when we get into the aging process. You know, you look at aged cheese, natural cheese. Similar, natural cheese was pretty flat prior to COVID. At that, you know, the first 3 months of the year, 74% increase, and cheese held because people were cooking at home at dinner versus going out for restaurants, and that 14% increase on a yearly basis. And then you talk about another thing is shifting how consumers buy the product. So historically, you know, people would go and walk into the store. You know, in the US, less than 25% of consumers had bought groceries online, and whether to drive and pick up or just to have delivered to their house. When COVID hit, over 55% of the consumers bought online, and so they used new delivery methods and new pickup methods to do that. So again, the industry is like, you're seeing people order maybe more gallons and go into the stores less. So it changed, and again, a lot of the packaging pressure and production pressure in the system. So when you look at e-commerce, you know, we were averaging prior to COVID, you know, on the dairy front, you know, around, you know, 50 to 70%. Post-COVID, over, you know, 3 times growth behind that. And then hunger, you know, looking at the hunger channel, and you see here in the US around 15 to 20% of food had been consistently food insecure, which is never a good thing. But when COVID, you saw dramatic segments of the population all of a sudden get impacts. I'll talk about that, what that did on the next slide. is, you've got 37 million households, and we've got programs here from, we call it SNAP, and then our food banks are set up, and they're set up to handle, weekend distribution and things like that on a national basis. And I think there's about 90,000 food banks in the U.S. COVID, again, a 17 million households. So, what is that? Close to 40% increase in demand. Just in last year, that resulted in 1 billion pounds of additional fluid milk demand through that system. So you think about where that last mile of food banks, it is usually distributed through, whether it's a church, a school, or a very small food center where they've got maybe 2 refrigerators, industrial refrigerators. When you got 1 billion pounds going through that system, it created chaos for them. The government was buying to help meet this additional food insecure. The industry was donating to meet this food insecure, but then the food hunger system didn't have the capability to do that. So there's a lot of partnering taking place to try to meet that demand. So just to put that in visual perspective, that additional 17 million people, this is the picture on the left is just how the food was distributed in the US. And so again, try to get healthy, nutritious food, and not just dairy, but think about produce across the US that's used to a much more normalized system. And so you had lines miles long to get food in the US, and it created a huge challenge to get nutritious food. The boxed, et cetera, only deliver so much health versus the higher levels of produce and dairy and other proteins. So this picture on the right is an example of what a normal, they may have 3 or 4 of these max at some of these food banks, and then yet they got an additional 1 billion pounds of just milk. That doesn't include dairy, that doesn't include the other produce that was bought. And so it just created a chaos in a system that wasn't really attuned to handling much mass refrigeration. And I think, with the government's looking at now, how do we build that last mile infrastructure for the future, and to meet demands for hunger system. And then talking about schools, the schools really, to at-home learning, created the need to, how do you get quality, the cold food that's distributed at schools safely to kids to go home and eat. So think about, a significant portion of the U.S. actually eats breakfast at schools and lunch at schools, what they were on a daily basis. And so we worked to help build systems within the school nutrition alliances in the U.S. to try to, everything from waivers. So like I said, daily the kids are getting the little milk cartons, to letting parents come in and take gallons home that will last them a couple days. So it makes it easier for the parents, makes it easier to distribute the foods. Number 2, on equipment, we've secured and worked with other agencies that we have relationships with to do equipment funding to provide things over here to the right, like portable coolers that they can distribute the milk to the kids to take home, and also like their lunches and sandwiches. And so there was the industry put not just dairy, but the whole, I'll call it the whole food secure systems. How do we make sure we're keeping youth fed? And in these major metros, if the schools aren't feeding, the kids have a lot of at risk. So we talked about 54 million youth in society that became food insecure. If the schools aren't providing the foods, there's a lot of concern about the kids getting the right nutrition to grow, et cetera, and to learn. So that's why it was so important as an industry across these areas, where we had excess foods in the food service world. We looked at, are there ways to repack some of those excess foods and or donate them to the hunger channel, or reprocess it for the schools channel, so we can make sure that a big portion of society is getting fed. So that's some of the key takeaways there. And from a total U.S. dairy impact, when we talk about the massive movement across channels, what's really interesting is, we still witness dairy consumption growth. I know my friends on the plant-based side like to tell the story that they're growing X%, but I said, when you got $100 billion category, 1% growth is $1 billion of growth. So it's just we're still seeing nice growth, and we've had historic growth both domestically and internationally. So I thought I'd wrap it up and just say, the growth came through massive and I'll call it partnerships to move products across different channels in a new way. And again, our focus was making sure these channels got food to those most in need, whether it's the hunger side or the youth side in schools. And then, retail and food service were more packaging systems that we knew would come back in the long term. So appreciate it. I'm going to turn it back over, and thanks for the opportunity just to talk about the hunger system and the channels.
Yeah, thanks very much, Paul. That was really excellent, and I think it's a nice counterbalance to the molecular and cellular science about nutrition, that in the end you've got to deliver foods that are nutritious to the population, and that's been challenging. So I think this was really excellent to hear some of the different the challenges and the strategies. Thanks very much. So Jeff, do you want to jump back in now, and the other two speakers, if you want to jump back in? And we've had a couple of questions have come in the question box there. I see a couple that are addressed to me, which are very common questions at the moment, which is really, is food enough, or do we need supplements for these micronutrients, particularly of interest? And if related to that, what about dosing? So I think in an ideal world, people would get the mix of things that their physiology needs, and that includes their immune system, from a diverse, well-balanced diet that provides plenty of plant-based foods along with some animal-derived foods to provide the things that the plants can't provide. Having said that, many of the studies where people have investigated the impact of micronutrients, for example, on immunity, they've all used supplements because that's easier to do in a randomized controlled trial. And they've tended to use intakes which are quite high, high compared to recommended intakes, and high compared to what might be achieved with most diets. This is particularly true for vitamin D, of course. So I think maybe, although food first is important, I think maybe to meet the needs for some of these micronutrients, modest supplementation might be required, and that's something to think about. Jeff, do you have a comment on that?
Yeah, so let me make a brief comment, and then I would encourage the others just to dive in whenever they want to add to that. So this is also related. I mean, we've talked about some of the incredible complexity of the immune system and immune response. But as we saw in the delivery, you also have this practical aspect of what can I do, or what can a population do? So even though we have so many studies showing that zinc is important, for example, measuring human zinc status is not easy. So we know that serum blood levels are not really reliable indicators of your zinc in your body. And then you say, well, okay, then my zinc level is like this. Should I eat a zinc-rich food, oysters? So I'm not being paid by the oyster lobby, but oysters are a good source of zinc. But then if so, how many should I eat? And these kind of basic questions, I think we cannot answer at this stage. So how do we translate some of those complex information and studies that we have into, let's say, things that people can do to prevent hospitalization?
Yeah, so I think the problem is that the science that provides the evidence base to answer your question hasn't really been done in the way that that needs to have been done. So, for example, we don't have good dose response studies for any of these nutrients, looking at meaningful immunological outcomes. And, you know, Paul de Vos talked about human studies that were set up to test hypotheses but failed to find results, or they found null results. That's very common, and I think this is the mismatch between you know, isolated observation and particular studies and the reality of the human situation. So I think we need better studies. Jeff, maybe we need to take into account that the system is more complicated than we give a micronutrient and we look for an effect. You know, we're talking about foods, we're talking about nutrient interactions, we're talking about the importance of the microbiota, all the other things that have been mentioned, you know, sex, age, genetic makeup, all those things. So I think it's very complicated, and you know, maybe a more systems approach to addressing this is a way forward, but that's not going to happen quickly.
Yeah, I think both Professor Fülöp and Professor de Vos talked about interdisciplinary actions, and I think you're right. I mean, just one example that comes to my mind, I think it's very rare when patients are admitted to the hospital for disease, not broken leg, but for disease, we don't take a nutritional assessment. So we don't know their nutritional status, and then we can't compare that to outcomes and see if that had the impact. I'm not even sure we could agree at this stage what to measure in terms of nutritional status. So sure. That could be one step that could help address that. This question also, so of course we always try to address, you know, what can I take to do this, and probiotics, prebiotics, and now even postbiotics are becoming an issue. So I know, Professor de Vos, you've done some work in that area with lactic acid bacteria and effects on immune system. Maybe you want to comment on that.
Yeah, I think many probiotic organisms will be very effective also to support antiviral responses. But as Fülöp said, the number of studies that had a design by which you really can focus on what the probiotic organisms or prebiotics have done on the immune responses are quite rare. You really have to think well about a design and really dedicate it to the ingredient that you are testing. And many of the vaccination models that we have applied, I mentioned it honestly, just failed because there was such a little area where you could have some improvement. We have to go to models where there is more space for improvement, so that you also can see transient improvements by the supplement that you give to the diet. And my feeling is that this is something that we really should focus on much more.
Yeah, thank you. And Professor Fülöp, so you've often talked about, again, interdisciplinary, and I think you've said at some point that there have been discussions, Alan Cohen, about even definitions of aging, and you've given very complicated aspects and the complexity. And I dare to say, besides the professors we have here, but I think a lot of nutritionists aren't so familiar with the incredible complex details that you've presented. And I don't know, but I'm not sure that all immunologists are aware of some of the insights of the nutrition side. And of course, we're all eating food, hopefully a healthy diet, to try to do that. How can that interdisciplinarity be improved and encouraged?
Oh, thank you. I think this is a very important aspect, and I am a geriatrician, so I see patients and I see even healthy persons all the time. And geriatrics, as you said, it's an interdisciplinary medical field. So in each of our team, a nutritionist and dietitian is included. So we are assessing the nutritional part of the subjects, but not the immune part. So we don't know whether what we do in the nutritional part have any connection with their immunity. So what we know that most of the time to intervene with just one thing, as in aging, as in everything, is not very useful and cannot be achieving complex changes in the whole system, which means that we always advocate if you don't have any specific deficiency in nutrients or in some nutriment, macro or micronutrients, you don't supply. So the only thing that what we try to promote in our older subjects is a very equilibrated, balanced diet. And we would really, as a geriatrician and also coping with aging science, to emphasize that what has been said, that it should start very early. So if you try to make changes, you can. Of course, you can always improve something, but sometimes when you start it at a very old age, it is not so efficient than if you started in middle age or even before. So this is very important. This is a lifelong process. And when we intervene, like for vitamin D, which is very, very important, because we see that most of the patients that we admit, and we measure this systematically in all our subjects, this is deficient. So we are supplying them, but we don't know exactly when we re-equilibrated their vitamin D system, what would be the real effect on their whole functioning. So, you know, these are questions that we should ask. And as you said, Professor Calder and also Professor Voss, that we need studies where we can assess the complexity in a system biology ways, the whole nutrition and the whole immune system and its interactions. But in geriatrics, we are very, very aware of the problems of nutrition, and we try to supply or even advise all our subjects to make differently or change their dietary habits.
Okay, that's very interesting. So I can mention that ILSI is looking at how we could address some of these topics, and we have a kind of framework of the kind of research to be done. But when you start getting to the specifics, of course, it's very complex. Which things to start investigating first to look at some of these fundamental questions. So that could be really useful to have some of those interactions. And as you say, we should always be looking at a healthy life. Personally, I definitely want to stop aging, but unfortunately, I know a lot of ways to do it, but they're all fatal. So we want to, you know, if you stop living, then you're stopping to age. But that's not what we want to do. We want to have healthy aging and try to encourage that. Let me switch gears a little bit to Mr. Ziemnisky's presentation. So the disruptions were very clear. And my question is, what steps do you think can be taken to be more adaptable in case of future? So there's always a risk of future pandemics. What steps do you think can be taken to prevent as big a disruption as was seen in the U.S. Food supply, especially liquid and perishable products?
I think two key things. One, the U.S. supply chain, having worked on, I'll call it the consumer packaged goods side, you know, we've squeezed our plants to the most efficient scale, you know, is it at the expense of flexibility. So I think a lot of people are revisiting flexibility. The other piece is looking long term of I'll call it new industry partnerships to optimize, you know, the different channels. And so there's a lot of very focused folks, and they can go in and because they're I'll call it cost driven. And I think if you put the consumers first and that consumer mindset, you can build in some of the flexibility and operate that way. So I think you're seeing an industry revisit just from a cost and throughput perspective to how do I have flexibility in my operating model, not just for pandemics, that's rare, but also just as I think about the different channels, more of a channel focus and flexibility. And also, I think, a better understanding of these global food systems of how do I meet, like, you know, the global food system needs. And so there's this reconsideration of just immediate sales versus I'll call it these long-term relationships and opportunities. And as you guys talked about, to the nutrition front is, we are looking at, how do you educate society? Their first choice today is they don't go to their doctor, they Google everything, right? And so you talked about, are they over consuming too many nutrients in one area and not getting nutrients enough? And who these claimed experts, are they really experts on the food education world out there, that an Instagram celebrity? And so when you look at our education system, for whether it's pediatricians, physicians, we're having a challenge of and say, do we need to invest more in the education in the nutrition space, like you said, and the future technology that you guys are discovering, and how do you to bring society up to speed on that, to make sure we're getting the right levels of nutrition? And because there's so many people that claim to be experts in society out there now, that you've got mass consumer confusion. And as a marketer, consumers are struggling with who do they trust for any sort of news today, right? You look at the Edelman Trust Barometer, trust in all kinds of sectors are down right now because you just had battles against each other. And so I think that's penetrating down into the food system, and it's scary because we want consumers to understand and trust what they can actually eat and what the right types of food sources. So you see this great work that you guys in industry are doing on science, and I think linking science to those who communicate to the consumer the value of getting the right types of foods, importantly critical.
Okay, thank you very much. So we're a little bit over time. We're going to have to wrap up, but let me give one last question, and then maybe people have one last comment. So I think Professor De Vos talked about this issue of machine learning, artificial intelligence. So it seems like that artificial intelligence is going to solve all the world's problems, including how we colonize Mars. But if you just bring this back to nutrition and immunity, the role of artificial intelligence and machine learning, just a brief comment would be interesting. Professor De Vos, maybe you'd like to start?
Actually, it was not me that spoke about that. I do not know much about artificial intelligence and nutrition. I think it was.
Okay, I thought you mentioned that you'd done some studies using machine learning.
I think it was Tamas. I might be wrong.
Okay.
You were imagining it. Yeah. You were imagining it. But I think there is there's a lot of data out there, and of course a lot of new data can be generated, and I think there is a role for machine learning in helping guide the way we might do our experiments, the things we might test, the things that are most likely to be effective. In part using existing knowledge, although that has got holes in it.
Okay, Professor Fülöp, any last comment on that aspect?
I think it can help us to make relationships, associations, interactions, but I think this help is a little bit overestimated. Because if you don't have enough knowledge behind and just trust only artificial intelligence, we can be misguided. So I think that this is a tool which shouldn't be overemphasized, but should be used to real human benefit, to increase the nutritional awareness and also the nutrition of subjects from the beginning to the end, to have a healthy immune system.
Okay, thank you. So I'll give all the speakers one last opportunity. If you have a short couple of sentences that you want to get a point across, please feel free. Or you think things have been covered?
Yeah, well, I think it's been a great symposium. We've covered a lot, but I think what we've really brought out is, the complexity of both the biological system and, in the last talk, the real life world in terms of delivering foods to people. These are all complex systems that, maybe COVID has been a catalyst to helping us optimize our understanding of these different systems so that things can be better post-COVID.
Yes, a lot of work to do. Okay, maybe we leave it at that. So I'd like to thank all the presenters and for the panel discussion and their excellent presentations. We could spend an hour on each individual presentation and even on some individual slides. So a lot of information conveyed very quickly, but a good discussion on an important topic. And I thank you again and all the participants who've joined us for this session, and look forward to seeing you again. Thank you all very much.
Yeah, I want to
Thank you really very much.
Co-chair of the session, the speakers, the participant, and just to let the audience know that the videos of the 8th session will be available to the 1,000 registrants in a couple of weeks, and the video of the talk will be made available to the global audience early July. So thank you very much again for your participation, and we're looking forward to welcoming you next year, hopefully in Belgium, for our next ILSI annual symposium. Have a good day. Bye-bye.
Okay, thank you.
Thank you very much. Bye. Thank you.